Hi, my name's Ginger Bonin, and I'm a biochemistry major here at Syracuse. And today we'll be talking about Sanger sequencing, which is this really cool method of essentially finding out what the exact nucleotide sequence of a DNA strand is. So we start with our double helix of DNA that we all know and love. You've seen it in movies and in all that popularized science. And we come through with a DNA helicase, which is just an enzyme that'll unzip it for us and let us kind of access these bases right here. And once we have this single strand after it's been unzipped, an enzyme called the DNA polymerase comes through and it grabs all these little T-shaped bases from around it and it pulls them in and it just kind of creates this strand back here that's going to be a replication of the DNA strand. And so the alteration that we have to make in sequencing if we want to know what the whole DNA sequence is instead of doing our T-shaped bases, we have what we refer to as bad bases, which are more of an upside down L shape. And the T-shape of the normal bases, it allows it to bind to the nucleotides next to it, because it's kind of like reaching out your arms and grabbing hands of people next to you. It can grab onto those nucleotides next to it. But if there's a bad base, it only can make one connection on one side, and then nothing else can bind over here. So what happens with the polymerase is it goes along, and it'll put down a normal base, and a normal base, and a normal base, and then it just randomly grabs a bad base, and then once the bad base is on there, if a normal base tries to bond again, it'll get kicked off and it won't be able to bind. And this does this randomly about the number of bases. So you can have 3 bases before a bad base, you could have 4, just like that. And then it's this difference in numbers of bases that gives the DNA sequence. Because what we do is we take all of our replications and we run it through a gel electrophoresis, which is kind of just like a science jello with electricity running through it. And this separates them out by length. So our shortest one is going to be an A, and then have adenine at the end. And then the one base longer is going to be a T, and then another base is going to be a G, and then a C. And since these are all just varying by one base, it'll give us that exact sequence of the DNA, and it'll keep going for the rest of the strand. And so why is this important? I mean, these are small little molecules that we can't even see. This helps us in our everyday lives. It helps us understand viruses and mutations. It helps us perform paternity tests, and it helps doctors diagnose diseases. It even helps us understand evolution, which is from millions of years ago, but these methods that we have now are helping us look way, way back into the past. And it helps us make better prescription drugs that help us with treating those diseases and those viruses. And so this is something I learned about a couple years ago, and it still gets me so hyped up about science because there are so many applications and the process is so cool. And this is why I decided to be a biochemistry major and study this kind of stuff with my whole life.